All right, so blood typing, okay, or blood grouping. Now, this is the old way we used to do blood grouping. Things have changed over these centuries. It's supposed to be a joke, but I guess it's just early in the morning and you're just not getting it. This is a bad class. This is bad. All right. Okay, anyway, blood groups. Now, there's a lot of different blood groups out there, but the more common ones that people hear about is what we call the ABO blood grouping. You hear someone's got blood group A, blood group O, that kind of thing. And also the RH factor, or the D. So those are the or the two most common ones that we have. But there are a lot of other ones, like 30 different ones. Now what it's saying is, we got blood groups M and N and Duffy. Not so concerning. We usually are more concerned with that with if mom has this, what does the baby have? And they're going to maybe need blood transfusions. All right, so that's what that is. I'm not going to ask you about the M and Duffy, and if the child has like Kell or Lewis or Duffy. Those are probably the three most common outside ones. But I kind of came up with mnemonics, and usually if the child has Duffy or Kell, then I usually say that the kid will die or get killed. Does that make sense? D and K. Whereas if you have Lewis, it's not something to worry about, so that one lives. All right. These two, I'm not saying they're going to die, but the thing is, these are the ones we have to be very aggressive and give blood transfusions while inside the mother's utero. This one we have to aggressively do. Lewis, we're not so concerned about. Okay. I know it sounds pretty gruesome, but when you come up with mnemonics, don't worry about what goes in your head and someone thinking about, geez, this guy's some psycho or whatever. Whatever is going to go in your head to make you remember this stuff, that's all that matters, right? So people wonder how I could remember so many different things. I usually do something that's I'm more of a pacifist. I don't really like violence and stuff. So to me, that's ridiculous. So putting ridiculous things in my head makes me remember them. Does that make sense? I used to do seminars for memory, and that's usually what works. All right. If I just use something that wasn't so ridiculous, I know I can't think of something, but I won't remember it. So I usually pick things like profanity in my head, or or let's say any kind of violence in my head. That's ridiculous. So I usually remember that. And I think you know what I'm talking about if you do cranial nerves and you came up with your own mnemonics and stuff. You want to think of ridiculous things.
Don't worry about it, you've been labeled now.
Yeah, I realize I've been labeled. Oh, jeez. All right, but it works. Trust me. I've done memory seminars, and they do work when you think of ridiculous things. Okay. All right. So anyway, blood group. So we have. Let's deal with the ABO blood group, and then I'll introduce you to. And once you understand this, which you will, then we'll throw in the RH factor. Okay. So let's talk about the ABO. Now we have antigens and antibodies. This will help you, if you understand this part now, it's going to help you when we get into immunology with your lecture. An antigen, if this is a red blood cell, an antigen is any marker. Now if I ask you to come to my house, and on my house, it's green and there's a white picket fence around it. And every house on my street is green with a white picket fence around it. You won't know which house is mine. Okay? It's just, and if there's like 100 houses, you won't know. So we got to put markers on houses so that we can decipher one house from the other. Whether we put a number on a house, whether we put a pine tree in the house, or windmill, or a different color like red. There's markers so that we can decipher one house from another. And cells have markers too. They're called antigens. So we could have no antigens on here. We could put an A antigen on here. We could put a B antigen on here. You could have just an A, you could just have a B, you could have an A and a B, or you could have no antigens. If there's an A, we say the blood type is A. If it's a B, we say that there's a blood type B. If it's both, it's AB. If there's none, we say it's O. All right? Do we understand what antigens are? Okay? Now, we also have antibodies. Now, antibodies will be floating around in your bloodstream. They look like little letter Ys. And they'll be floating in the bloodstream. The antibodies will correspond with antigens. There's actually a variable spot here that will actually mimic or fit like a locking key on one of these antigens. So this could be antibody to A. This could be an antibody to B. If we have a B antibody with a B antigen, they will come together, set up a chain of reactions, and destroy that red blood cell. You don't want an antibody and antigen corresponding to the same letter in the same blood group. These can live in harmony. These cannot. Okay? And we'll talk about that. All right? So we say that this is anti-B or antibody B. This is antigen B. When these come together, they're going to actually bring more red blood cells that have B all together over here. They're going to what we call agglutinate, not coagulate. Okay? Or, I'm sorry, or agglutinate or clump. All right? Not coagulate, not clot. That's different. So the antigen will bind to the corresponding antibody. They'll connect. Red blood cells with similar antigens will agglutinate together, form this lattice appearance. A chain of reactions occurs, which you'll learn about in immunology, and will destroy those red blood cells. We say that's hemolysis. Hemo meaning blood, lysis to break up. Okay? So here you have red blood cells with a certain antigen on it, the blue here, in correlation with the same antibodies, or at least the corresponding antibodies that fit like a lock and key on those antigens. If that should happen, the red blood cells will agglutinate, they'll clump, but not clot. Please don't use the word clot. That's platelets, and students use that, and that would be wrong. So I like to use the word agglutinate. It's a big word, I realize that, but it'll keep you from saying the word clump. Okay? You could use clump, that's correct. A chain of reactions occurs, and the red blood cells will be destroyed. Right, or antigen A and antibody A.
But if DNA won't ever connect.
They won't ever connect. Okay? So far so good? Okay, now, you put your pencils down. This is probably the most important slide of the whole presentation. There's so much information on here. Okay, but it's nicely put in a schematic view. Forget about these over here for the time being. Let's look at this one here. Here's a person who's type A, very stressful, blood type A, okay? On their red blood cells, they will have antigens to A, represented by these balls, these yellow balls, okay? In their bloodstream, they will also have antibodies to B. Now, they can live in harmony. The antibodies to B have almost like a triangle kind of shape over here. That's, again, it's just showing schematically. It's not real life, but it's so you can visualize what's going on here. These triangle-shaped patterns don't fit onto the balls there. So these can live in harmony. Antigens to A, antibodies to B. That's what happens in a person who has A blood. Okay? Questions on that? Okay. Now, let me do something. I forgot to show. Let's just do this as a fast review because we're going to get back to this. We take blood from someone's arm, we put it in a centrifuge, or we put it in a test tube, we spin it around for about 5 minutes, and all the heavy stuff goes on the bottom, all the light stuff goes to the top. This whole thing is called what? Whole blood. You guys studied, that's good. or just easy? I'll challenge you, don't worry, okay? All the heavy stuff is gonna be down here, which is the majority of the stuff. What is this? These are the red blood cells. And this makes up about 45% in a healthy person. 45% of the whole blood. Then you're gonna have this area here, very thin area. And what is that gonna contain? The white blood cells and the platelets. That's gonna make up about, well, less than 1%. And it's also known as the buffy coat, or buffy layer. Because it looks white. When you look at it, it's actually white. Then you have over here, mostly water, but all our hormones. What is that? Yeah, that's the plasma. And that makes up approximately 55%. You'll have mostly water in here. 91% of the plasma is water. The other 9% is going to be things like your oxygen gases, your antibodies, okay? Other proteins are made of proteins, so you have other proteins in there, albumin. You'll learn about all this kind of stuff. All the hormones that are in there, certain enzymes might be in there. So it's all that kind of stuff, okay? Now, let me explain to you before we get back to this, that when you give blood, you're given whole blood. You're not just, you know what I'm saying? They're not just going to take red blood cells out of you. It's impossible to do that. You're taking whole blood out of you, they spin it around, and then they can separate it. Now, in the blood bank, what they do is they look at the shelves up there, and they see what they're low on. If they're low on whole blood, then they're just going to take your whole blood, put it in a bag, and put it on that shelf. But we could actually split this up. Once you centrifuge it, we could actually then extract just the red blood cells. We could just extract the plasma, okay? We could extract the platelets. White blood cells we don't usually give out, okay? It's kind of different because they have a nucleus and they can multiply. We don't do it. We do bone marrow transplants to do something like that. But you could separate this, and if we're low on platelets, then we'll put it up there. You can imagine we need a lot of people to put platelets on a shelf, all right, because you only have a very little amount. So we'll put that up there. If we're low on plasma, we'll put that up there, okay? We only give you whatever you're missing if we got to transfuse that. If you're low in red blood cells, for instance, you got sickle cell anemia, the red blood cells are deformed. So we're only going to take out the red blood cells and put in red blood cells. Does that make sense? Okay? If you're low on plasma, then we're just going to replenish you with plasma. If you're on your menstrual period and you're losing like everything, sure, we're going to give you iron tablets because you're losing a lot of iron, but you're losing everything. So we're going to give you whole blood. All right? Now, I don't want people to think that, oh, I'm clotting it. No, I'm saying if it goes down like a hemoglobin, it goes down pretty low, you know, and you're pale, and as soon as you get up, you faint, yeah, then you're having symptoms. Of course, we'll then be aggressive with giving you blood transfusions. But we only give you what we need to give you. Does that make sense? We don't give you stuff that you don't need, okay? Because we can savor the other stuff for people for what they need. Now, again, just to reiterate, this is blood type A. They have antigens to A, represented by balls, yellow balls on here, and you have antibodies to B, which kind of have like a triangular pattern on here, that will not correlate with the balls on here. So these two, antigens to A, antibodies to B, can live in harmony. There's no issues with that. Are we okay with that? Okay, now, let's look at a person who's blood type B. What they have on their red blood cells is antigens to B, which kind of look like these pointy yellow things. In their plasma now, they have antibodies to A, which have like a curved kind of pattern here. Again, it's schematically, it's not really curved and it's not really pointy, but they're doing it so you can see it. These can live in harmony. Antigens to B and antibodies to A. They won't agglutinate. Does that make sense? Okay? Now, let's give you a little twist here. Here's a person with blood type A. We want to give red blood cells of B to a person with blood type A. What's going to happen? The antibodies that's in the recipient of B, is going to correspond to the antigens here. So as soon as you're going to get that red blood cell with B, it's going to get destroyed. It's useless. You might as well just take this blood and pour it down the toilet bowl because it's useless in that person. Okay? Likewise, if we give red blood cells of A antigens to a person with B blood, this will correlate. Does that make sense? And it'll set up a chain reaction to destroy it. Now, here's what's going to happen. Because blood goes throughout the whole body, the reaction happens throughout the whole body. All right? This is what we call a blood transfusion reaction. It's not just going to happen at your arm where you're putting the blood in. It's going to take a few minutes as the blood circulates throughout your whole body. The antibodies are going to see that and destroy it, and now your whole body is going to go and react to it. Because of this, it goes into your brain, it goes everywhere. You'll have what we call an anaphylactic shock, and you will die from this. A blood transfusion, given the wrong blood type, you can die from this. Does that make sense? Okay, because it's going through the whole body. Person with AB blood. They have antigens to A and B. In their plasma, they wouldn't have any antibodies. Does that make sense? Okay? Even if you have A antibodies, it'll correlate here and destroy it. If you have B antibodies, it'll correlate and destroy it. You don't need A and B antibodies to destroy this. You just need one or the other. Does that make sense? Okay? And a person who's type O, they don't have antigens to A or B. Okay? But they do have both antibodies. And that will live in harmony because this has nothing to bind to. Is that clear about that? Now, let's give you some scenarios. Can I give red blood? And by the way, when we give just red blood cells, sitting in the blood bank, we call it not just red blood cells, we call it packed red blood cells. All right, because they're packed together. So whenever you see PRBC, that's what they're called, packed. Okay, and you'll see that when you get into your medical realm. So if I'm just going to give packed red blood cells of AB blood to a person who is blood type A, can I do that? No. Why? The antibodies here of B will connect here and destroy them, and you'll go through a transfusion reaction. Okay? Can I give this blood, red blood cells, to a B person? Likewise, because the antibodies here will correlate and destroy them. But what about giving red blood cells of AB to an O person? Can I do that? No, I can't do that either because there's antibodies here that will correlate here. So the only people that that AB blood can actually be given to is AB. Can I give these red blood cells to an AB person?
Yeah.
Mm-hmm. What about these red blood cells, B, to this person here? Sure. There's nothing that's going to destroy them. What about this over here? Mm-hmm. This is why blood type AB is the universal recipient. They can take everyone's blood. And the reason why, and you should need to know this, is simply because there is no antibodies to A or B in blood type AB. That's why they're the recipient, they're the universal recipient. Okay? Before you were memorizing it, but You're in my class, I want you to understand more, okay? If I have this red blood cell here of O, can I give it to this person who's AB? Can I or no? Yeah. There's no antigens here that antibodies would correlate with. Can I give this red blood cell to a B person? How about to an A person? The reason why is because there's no antigens here. So type O has no antigens, so these antibodies have nothing to connect with, so you could give this blood type, this red blood cell, to an O person, of course, to an AB, to a B, or to an A. And the reason why this is known as the universal donor is because it has no antigens. Okay? Does that make sense? All right? A lot of information on here, but it really summarizes everything for you. All right? Questions on that? All right, so just to reiterate, if you have type A blood here, and you have antibodies to B, they will not agglutinate or clump. If you have A antigens here and A antibodies, they will clump or agglutinate, and it'll set up a chain of reactions and destroy these red blood cells. Sound good? Okay, now, let's talk about the experiment today, and then we'll do RH and I'll let you guys loose. There's a tray in front of you that kind of looks like this. All right? Have everyone grab one so you know what I'm talking about, okay? Now you're going to see some changes in here. We're going to put some liquids in here, and it's better to see the changes under a white piece of paper. So if you have a white piece of paper or something that even has words, it'll become blurry. We're not going to do it just yet, but just to get prepared. The black tops on here, you just can't see those changes properly. So if you have a tray with 3 wells on it, and it should read something like that, right? A, B, and RH. Do you have that? Okay? On the same page? Now, let's forget about RH for a moment. In fact, let's forget about B for a moment. Let's just talk about this well here. Now you have, I also put on your table, we have these liquids, okay, different color liquids. What these are, it'll say anti-A, anti-B, anti-RH. These are actually liquids that contain antibodies to that corresponding letter that's on the bottom. So anti-A, this is antibodies to A. And what we're going to do, not now, because it'll dry up if we do it now, but what I want you to do when we're ready, we're going to put 3 drops of this serum, so to say, into the well of A. What we're putting in the well of A is antibodies to A. We're putting those little Y's, and we're putting them in there. Okay? You'll also put antibodies to B in here and antibodies to RH. But let's just consider this one over here. So right now you have 3 drops of just antibodies there. Then we have the simulated blood. We used to do real blood, your own blood, but the bodily fluids, we can't do that anymore. So we have the simulated blood, which is okay, but the real blood is, it looks better. So we have the simulated blood. In each one of these bloods, I have like a few different ones on here. It'll say Mr. Brown, or it'll say sample A, sample B, or sample 1, sample 2. So you don't know what blood type this is. Okay, it's like the way you have, you don't know what your blood type is. So what you're going to do, you have antibodies to A here, and then we're going to put, that was 3 drops in here, and we're going to put 1 drop of blood, unknown blood, in here. So you have these antibodies to A that are just floating in there. And they're looking for something to attach to. What are they looking for? Antigens to A. That's all they're looking for. Anything else they're not going to be looking at. So you're going to put 1 drop of blood in that A. And lo and behold, in this particular instance, when you put a drop there, you're going to see kind of like that. Because what happens here is that the antibodies to A, they're looking for that antigen to A, and you put a drop of blood in there, whoop, and it sees an antigen to A. So it goes, wow. It sucks right up there. And when it does, the blood will agglutinate or clump, and it'll look similar to that. You can see it up here. If that happens, then you know for sure that there's antigens to A in that blood. You can't say anything about B or RH. You can only speak about A because you're only working with the A1 here because you're only using A antibodies. Does that make sense? Okay? Now, you put 3 drops of antibodies to B in the well of B. So now you have B antibodies floating there. You put the same blood, 1 drop of the same blood that you put here, you're now going to put it in here. Clear with that? You put a drop. Here's the antibodies to B. Bloop. What is antibodies to B looking for? Antigens to B. But it doesn't see it there. So they're not going to clump. Instead, it'll just look like this. So you could say that there's, in this particular instance, you have antigens to A, you don't have antigens to B. So right now, you could say that this person's blood is type A. Don't get the positive and negative. I know people think of, well, that's O negative. We'll worry about positive and negative later, okay? That has nothing to do with the ABO blood grouping. So right now, we can say that this person's blood type is A. In a person with blood type A, in their real blood, what antibodies would normally be found in that blood? B antibodies. Good. Catching on? Okay. So this is just, you could try to memorize this, but I don't like to memorize this. If you understand the concept, who can give blood to and who can receive it, understanding it is better than just memorizing. But I put this up here so you can actually see that. Okay? We could also do blood typing. Okay? With the good old Punnett squares, right? You guys have done Punnett squares, right? Fun stuff. Okay? They're the scientist version of Sudoku. Okay? So this is what's going to happen. This is mom. We abbreviate the symbol for female as that, and male will be over here. That's a symbol for male. Now you could get an allele from mom and an allele from dad, right? So if you, and we use the same thing here. So you could get, this would be, you got an A allele for mom and an A allele for dad. So therefore, this person's blood type is A. 2 dominating ones. Okay? You could also get an A allele from mom and an O allele for dad. The O is actually recessive. Right? It's kind of like you've seen this before, right? We're just using different letters. So this and this means that it's going to be whatever the dominating H is. Does that make sense? Okay? We're just using the recessive is O. So this is going to be A. So the blood type will be A. The only way you could get O blood type is that you have to get an O allele from mom and O allele for dad, and then you'll be O. Does that make sense? Okay, how to read these? So if mom here is, let's say, blood type A and B, and dad was O, then we just do this whole thing. And the offspring, 50% will have A blood type and 50% will have B blood type. Does that make sense? They won't, none of them will have AB and none of them will have O. All right? If they have O, the father will be saying, well, yeah, I'm a type O, so my child's a type O. This is the stuff you see on Maury Povich. Whatever, I'm the father, look at that shit, you're not the father. You can't be, it's impossible. Does that make sense? This is a quick paternity test that you can do. Now, sometimes you can't tell, you know, if let's say the blood type is B, that doesn't mean that the father, that's the real father or not. But if it's type, it couldn't be. It's impossible, right? And that's the kind of things you see in, I had this finger up. I'm recording, it's this finger. But that's what I'm saying, is those kind of things over there, okay? Questions about this? So 50% will be AO and 50% will be BO. Well, not BO, but, you know, right? Okay? So those kind of questions might appear on your lecture exam, how to work those things. All right? And this you won't have to memorize, but it just gives you an FYI about which ones are the most predominant in our population. All right, just for curiosity. What's really amazing about this is that O blood type is the most common, and yet it's recessive. It's kind of a weird phenomenon that happens. All right, AB is the most is the rarest of them all. Okay, so 45% of the population, or at least the white, they broke it down to whites, blacks, Asians, Native, but they're just, it goes across the whole thing that O is the most common one. It's kind of weird. I think it's weird that, you know, recessive is the most common. Okay, you would think that that kind of weeded out, and I don't know what the rationale of evolution, why that happened. Okay, so it's that. Now, let's talk about RH blood group. Okay, now that you understand this, RH is going to be a piece of cake. Okay, remember how I was explaining a red blood cell could have an antigen to A. There could be an antigen to B, but we could also have another antigen, and it's called D. A lot of times we'll call it the RH factor. It doesn't matter to me if you call it D or RH, okay? There is no C. I guess the same way why there's no B batteries. I have no idea. Makes you wonder about whatever happened to preparation A through G. It makes you wonder, right? Maybe preparation C was that the whole butt lit on fire. Well, we can't use that one. Let's go to D. You know, that kind of thing. Preparation H is for hemorrhoids. Okay, so we could have another antigen here. So we're just adding another one. That's all we're doing. Okay. Now, 75% of the population has the D antigen, the RH factor, and 25% of the population does not. If you are born with the RH factor, you will die with the RH factor. If you are born without the RH factor, you will die without the RH factor. You don't gain it anymore. Okay? Does that make sense? Now, the best way to explain how this works is a disease called Erythroblastosis Fetalis with pregnant moms. Okay? Now, before I get into this, I need to explain to you that the baby's blood and the mother's blood, with baby meaning like inside the fetus, okay, so inside the mother's womb, the fetal blood and the mother's blood never, ever, 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 ever mixes. Meaning, I'm not talking about the gases or let's say viruses and stuff. I'm talking about the red blood cells. The baby's red blood cells doesn't go into the mom. The mom's red blood cells doesn't go into the baby. Never, ever, ever. Except, okay, 2 times. One, if the mother who's pregnant has a trauma that happens. She gets in a motor vehicle accident. And now that placenta, if this is where the baby is over here, if that placenta shakes and moves, now there's going to be some blood from the placenta from baby going into mom's. and vice versa, okay? Or mom tumbles, or there's domestic violence and, you know, she falls down or gets hit or something. You see what I mean? If there's something like that, yes, blood can mix during that time. The other time, which is all the time, is when you deliver the baby. The baby comes out, and now we're going to remove the placenta. So now as you remove the placenta, take that placenta off the uterus, there's going to be a mixture of blood happening there. So those are really the only two times that this happens. Also procedures, like amniocentesis and stuff, there's a possibility that it can go into the placenta. But those are really the only things. It's something invasive for that to happen. It doesn't just happen just all of a sudden, okay? So, we're assuming that mom and baby don't have a mixture of blood. Now, here's where the problem is. Let's have it where mom is Rh negative, meaning she doesn't have the Rh factor on her blood, but the fetus or the baby does, because the baby got that from dad, and that's okay. So what's going to happen is, if the fetus is Rh positive, mother is Rh negative, and then we do have a trauma, and now the blood is going to mix. So baby's blood is going to go into mom's blood. Mom sees a foreign thing there, Rh factors, never seen it before. And your immune system will take effect in antibodies to attack that. So here you have Rh factor coming into mom, foreign thing. Mom is going to make antibodies and will attack that. Now, the amount that goes from the baby into the mother is very minute, a few drops. And because of that, the woman's body does not go in an anaphylactic shock. She doesn't die from this. In fact, she won't even notice that this is actually happening. But it doesn't matter. She is making antibodies against that, destroy those few, and then what happens is, and all you are is a collection of antibodies throughout your entire life. Those antibodies that she just made stays with her for the rest of her life. So that if another antigen with Rh comes in there later on in her life, she already has antibodies that could actually go much faster and destroy it. That's how chickenpox works. You get infected with the chickenpox virus. It's going to take 2 to 3 weeks for your body to actually make antibodies and destroy them. That's why you got all the itchiness and all the vesicles that are on your body for chickenpox for about 2 to 3 weeks. But once your immune system builds up, it'll destroy that, make the virus hide someplace else. But the thing is, it destroys it, and now if you ever get exposed to chickenpox ever again, it happens so fast that nothing actually happens to you, because you have those antibodies that you've collected. Everything that you touch, everything that you expose your body to, you're making a new antibody. Babies don't have many new antibodies, obviously. They only get whatever mom gave them, a certain antibody that crosses the placenta. That's it. But gradually, they need to do this. That's why they get a lot of vaccines, okay? But you have a large creation of these antibodies. And you'll learn about that again in immunology. So let's go back to this. So baby's blood goes inside mom. Mom sees this foreign thing, antigens to Rh, and she makes antibodies, destroys them. Mom is doing okay. Baby's doing okay too. All right, baby came out and baby's doing fine. There's nothing wrong with that. Now, are we okay with that so far? Okay, now, mom gets pregnant again. She gets pregnant again, and she's still Rh negative. She doesn't have antigens, but the thing is, she also now has antibodies to Rh. And if she gets pregnant with another baby that has an Rh factor on its antigens, if there's another trauma that goes on, and baby's blood goes into mom's, or mom's blood goes into baby's, Mom's blood is going to have antibodies to Rh, and it goes into the baby's blood. Those antibodies are going to find those antigens on baby's blood and destroy them. That's not a drop of blood. That's all of them. And the fetus is now going to go through an anaphylactic shock. You don't usually see it that way, but as soon as a red blood cell comes out, it gets destroyed. They get severe anemia, and then they'll die from that. So it's not the first time, it's the second time that we're concerned about, because mom has these antibodies. So on the second pregnancy, babies, if the babies are, you wouldn't know. You're not going to test the blood on the baby until the baby comes out. So we're assuming the baby has Rh positive blood. Mom is Rh negative still, but she has the antibodies to Rh. If they go into the fetus, it's going to cause hemolysis to the red blood cells, destroy them, severe anemia, and that'll cause death. This is a disease called Erythroblastosis Fetalis. So, how can we prevent this? We got to find a way, because there's always a possibility if mom is negative, Rh negative, and baby's Rh positive, there's always going to be a possibility that some of that blood is going to go into baby. You can't change that, okay? It's always going to happen, or not always going to happen, but there's a possibility it can happen. So what we could do is find some kind of measure, or some kind of treatment that we could prevent mom from making those antibodies. Then you never have to worry about this, right? Well, we found something in the 1960s, and it's called RhoGAM. RhoGAM is an injection that we give with any kind of possibility that blood, the baby's blood can mix with mom's or vice versa. We will give, and mom is Rh negative. We don't give this to moms who are Rh positive. There's no reason for that. But if mom is Rh negative and there's some kind of motor vehicle accident or procedure like amniocentesis or even delivering the baby, we will give mom RhoGAM. And this will prevent mom from making antibodies. Now obviously if the baby comes out, we'll test the baby. And if the baby's Rh negative, there's no reason for RhoGAM. But if inside the mom, we're not going to test the baby if it's inside the uterus. This is a little one-shot thing, a little $20 kind of thing. We're just going to give it. There's no harm with the baby. There's no harm with the mom. We'll just give the RhoGAM. And that's what happens. Anybody here Rh negative? And have you had a child before? Or? Okay. I expect that you'll probably get all these shots because you're Rh negative. Nothing bad. It's a precaution to prevent this erythroblastosis fetalis. Okay? Questions on that? Yeah.
I knew like my husband was Rh.
Right, then you wouldn't have to worry about it. If he's Rh negative and you're Rh negative, yeah. And I'm not saying with you, but that's where the Maury Povich kind of stuff comes in. Not, I mean, if you know you're in a monogamous relationship, then that's fine. It's just that, you know what I'm saying? I don't want to get too much in that, you know, that kind of thing. But yeah, but we don't know. We want to always test the father too to make sure that's it. Mm-hmm. Now the other thing I just want to also mention is that I've been talking about babies, fetuses, babies, but it's wrong just to be thinking that way. It's any pregnancy. If you had a miscarriage and you're Rh negative, we're not going to test the products that came out. So we assume that the fetus or the embryo was Rh positive, so you'll get RhoGAM for that. If you had an abortion, if you had an ectopic pregnancy, we're not going to test that products of conception, all right, the pregnancy, so to say. So if you're Rh negative, we will also give you the RhoGAM for that stuff. Even if you think it's the first pregnancy, it might not have been your first pregnancy. You could have been pregnant before and not even realized it because maybe it was just a different kind of discharge, maybe a very light discharge, and you had a miscarriage and you didn't consider it a miscarriage. It was just you know, I just was clotting a little bit heavier that day. Does that make sense? So we give it no matter what if you're Rh negative. Questions on that? Okay. So this is in a summary, putting it all together. But if mom is Rh negative, baby's Rh positive, We deliver the baby, but some of baby's blood is going to go into mom. So mom's not pregnant in this picture, this stage here. But baby's blood, which is the pluses over here, went into mom. Mom is going to make antibodies to destroy those red blood cells. That's what this circle plus is. And now mom has antibodies for the rest of her life. If she should get pregnant again and there's Rh positive in that baby, there's a possibility, for whatever reason, her blood can go into baby's blood and destroy the red blood cells of the baby, and that will cause death of the baby. Does that make sense? Okay? All right. So, let's now throw in the other thing. It's the same thing when we're dealing with this. Rh antibodies. We're going to, whatever the, yep, I think it's clear, colorless. You're going to put 3 drops of antibodies to Rh in here. And now you've got antibodies to Rh there. Whatever blood you put here, whatever blood you put here, you're now going to put a drop of that same blood in here. Rh antibodies are there. You're going to see clumping. In this particular case, you'll have to see clumping. What can you tell me about antigens in this well only? There is Rh antigens there because the antibodies cause clumping or agglutination. Does that make sense? Yeah? All right. So now, let's put all 3 wells together. What if I came up with this? Okay? And 3 questions I can ask is what person's blood type this is, what antigens would be in this blood type, and what antibodies, and these are my abbreviations for antigen and antibodies, would be in here. So, you put drops of antibodies in here, and you see clumping. Is there A antigens in that well? All right, don't worry about the other antigens. I'm just looking at this well only. We're only concerned about A antigens. Are they there or not? Mm-hmm. So you're going to have A antigens there. Okay? Let's look at this one. Okay, you put antibodies of B here. There's no clumping. Is there B antigens in this well? No. Okay, so you wouldn't write that there. You put Rh antibodies in here. There is this agglutination. Is there Rh antigens in here? Mm-hmm. So you would say that there's A antigens and there's Rh antigens, or D, whatever you want to call it. So the blood type in this person is going to be A and Rh, but we don't say that. If there's an Rh, we don't call it Rh, we just say, is it there? Yes, then we're going to call this positive. So this person's blood type is A positive. The positive is only referring to the Rh. Okay? In a person who has A positive blood, what antibodies would they have? Would they have A antibodies? No, it doesn't. It can't live in harmony. You can't have A antibodies and A antigens. So you would not have A antibodies here. Would you have B antibodies? Yes. In the person's blood, not in our experiment here, in the person's blood. Would you have Rh antibodies? No. So that's one practical exam station. I'm going to give you a blood type here, and then here's 3 questions. You should be able to do that in a minute and a half. Okay? Let's do another one. I'll give you a few moments. Do it by yourself, and then we'll go over it. Give you 2 or 3 minutes to do that. All I want to know is, this is what I'm going to give you. What blood type is this? What antigens are in this person? And what antibodies will be in this person? Okay? All right. Just a show of hands. Who thinks this is A positive? B positive? A negative? B negative? If you're too shy to put your hands up, just go like this a little. You don't have to go like this, and you're the only one putting that hand up. All right, just like that. AB positive? AB negative? O positive? O negative. Good, you got it all right. All right, so this is O positive. Okay? In a person who's got O positive, what antigens would they have? Would they have A antigens, B antigens, RH antigens? Yes, don't forget the RH antigens. They're there. In a person who's got O positive blood, would they have, what antibodies would they have? Would they have A antibodies? Yes. Would you have B antibodies? Yes. Would you have RH? No. And there's no such thing as O. I'll get that occasionally too on my practical exam questions. Yes, but they have O antibodies. There's no such thing as O antibodies. Okay? You're doing a trick. That's not a trick question. That's like a trick answer you're trying to give me. There's no such thing, right? All right, let's do another one. I really want you to get this down pat. Okay. Give it a couple moments and see if you can do this one. Again, I want to know the blood type, antigens, and antibodies in there. Come on, this is better than Sudoku, right? Okay, who here thinks this is O positive, O negative, A positive, A negative, B positive, B negative, AB positive, AB negative? Good. Most of you've got it. It's B negative. Not my model. B positive is my model. B negative. In a person who's got B negative blood, what antigens would be in there? Who says that there is A antigens? B antigens? Yeah, B antigens. What about RH antigens? No, good. And what about antibodies that's found in their blood? Would they have A antibodies? Mm-hmm. Would you have B antibodies? No. Would you have RH antibodies? Not in my class. Women get their ROGAM. Okay? They get the ROGAM and it prevents that. I guess and that's why, I mean, think about it. They started making the ROGAM in the early 60s. So before then, we had a lot of miscarriages. Once we got the ROGAM, it kind of cut down on miscarriages. Okay? I guess people who want to deliver their babies in the mountains and choose not to see a doctor or go to the hospital, yeah, I guess maybe those would, but for a majority, we just don't see the situation, you know, in case of those rare conditions. Okay? So we get the ROGAM. In my class, we don't have RH antibodies. We're moving forward with that. Okay? Okay,